Welcome back to Particle Pharma. In my previous session, I have talked about cell division and I have completed mitosis. So today I will be talking about meiosis that can be seen in germ cells or germ-line cells where a male and a female gamete fuse to form an offspring which is also having a 23 pair of chromosomes. As fusion is taking place, so as both are having 23 pair of chromosomes, so the offspring should also have 23 pair of chromosomes. But there is no increase in number of chromosomes. Instead, the number of chromosomes is same. Or if it has to be 46 pair, it is not like that. Again, it is getting reduced. So this process is known as reduction division. Next is why there is no increase in number of chromosomes from one offspring to another offspring after the fusion is there will be some reasons. First one is genetic imbalance. There will be a genetic imbalance which causes or which leads to genetic instability causing developmental defects. Next is species incompatibility. If there is an increased number of chromosomes for every division, then species won't be able to reproduce. There will be an inequality or the species can face some issues. Next, nature stability. Natures will get unstable, which leads to evolutionary consequences. There will be a formation of new species. Again, it will be a disaster for the evolution. So for these reasons, there is a decreased number of chromosomes for every division. Next is, so the cell before entering into a meiosis, first it should pass through interphase. As we have known that interphase is divided into three subphases. That is gap 1, synthesis and gap 2. In gap 1, there will be a formation of proteins, organelles, enzymes and molecules. Next, in synthesis, there will be a DNA duplication or replication. If everything goes well, then it will proceed to gap 2. In gap 2, there will be a check for error through checkpoint. They will check whether the DNA replication is correct or not. If it is correct, it will proceed. Otherwise, it will stop here only. And there will be a formation of spindle fiber in gap 2. So let me begin with meiosis 1, which is divided into prophase 1, metaphase 1, anaphase 1 and telophase 1, which is followed by meiosis 2. It means meiosis is divided into two phases or it is getting completed in two phases, meiosis 1 and meiosis 2. So prophase, meiosis 1 is starting by prophase, which is a longer stage or crucial stage. Why it is a longer stage or crucial stage, we will see in the coming slide. The prophase is, we can see there will be a pairing of homologous chromosomes will be there. Pairing of homologous chromosomes can be seen in this stage. How it will happen? That we can see in 5 different subspaces, that is leptotene, zygotene, pachytene, diplotin and diakinesis. Leptotene, their chromosomes started to condense. Here chromosome is not getting condensed. It means they have started to condense where chromatids is not completely visible. And nuclear envelope and nucleolus is still intact. Next stage is zygote, where homologous chromosomes are paired up, or that process is known as synapsis. So this homologous chromosome, where chromosome is getting condensed, now sister chromatid is also visible. So what it will do is they will come nearer to each other and they will start to share their genetic material which is present inside the chromatids. So not from the same sister chromatids. I mean, this is one chromatid and this is another chromatid. Wait, let me draw it properly. This is one chromosome and these are two different chromatids. This is one chromatid and this is second chromatid. This is another chromosome. This is one chromatid and this is another chromatid. What will happen in pairing in prophase 1 is the genetic material which is present on this sister chromatid. Okay, on this single sister chromatid, they will start to getting transfer on this sister chromatid. So this process is known as pairing or recombination, due to which we can see genetic diversity. That's why there will be different offspring. If they are sibling also, there will be difference in them due to this process. And this process, when this sister chromatid will come to this sister chromatid and they will start sharing their information or the genes which are present on that chromosome, or I can say allele, they will form a tetrad. Means they will come closer. In diagram I will show you. Like this it will happen. So this whole section is known as tetrad or bivalents, where four sister chromatids in paired homologous chromosome can be seen. So moving on to the next stage, and zygote is known as pairing stage. Okay. So moving on to the next stage, that is pachytene, where chromosomes are completely condensed or they are in packed stage, where tetrad formation, just now which I have explained, where tetrad formation is seen and crossing over occurs. They will come closer and the genes will transform from one sister chromatid to another sister chromatid of different chromosome. I repeat, the sister chromatids will start sharing their genes or genetic information from two different chromosomes of the same cell. Takes place in pachytene. Next is diplotin. Diplotin. So here synaptonemal complex dissolves. The complex through which sharing is taking place, that will get dissolved, but they will get attached at chiasmata, that is prolonged in female but cannot be seen in male. Male it will happen then itself, but it will be long. I mean it is an year process. Till ovulation they will be attached. Next is diakinesis or movement phase, where compact chromosomes or nuclear membrane begins to dissolve and nucleolus also disappear, as we have seen in mitosis also, where it is happening in the anaphase, sorry, telophase, where spindle fibers begins to form and it is preparing for next metaphase. This prophase is preparing to enter into the metaphase. I'll be showing in diagram. You see here. Spindle fiber, we all know that they are going to form and they will try to attach to and they will get attached to centromere, which is the center to attach to of different chromatids, or sorry, of same chromatids. That is the center. Next is chiasma or chiasmata, where two different chromatids share their genetic information, where crossing over takes place. And the chromatids of the same chromosome is known as sister chromatids, and the chromatids of different chromosomes is known as non-sister chromatids. Tetrad, these two different chromatids, I mean these two different chromosomes or pair of homologous chromosome is known as tetrad. So this is the prophase 1 of meiosis 1. In the next stage, I'll be, or in the next video or session, I'll be talking about the other phases of meiosis 1. Till then, stay tuned, try to be in contact with us, and if you find any discovery regarding the cell or in the science, please let us know and try to get the videos and try to see the videos on our Instagram page in highlights session. Thank you.